For joining us this morning. This session, as you see on the slide, it's supported by the Michael Horn Fund, and this is a fund that was created to really honor the memory of Mike Horn. If you didn't know him, he was a long-term member of the society, and the endowment was really started in the to really honor his memory. And Michael really was an active member of the society since 1990, and he was actually for a long time also the editor of the Tissue Culture, the Tissue Culture Association report, and he was mainly the editor for what was called the Explant. And he was a very innovative, and that's why this session is associated with his name, kind of scientist. And he was also very active in the society. He was the chair of the plant section from 1998 to 2000, and then he was back the chair of the plant section from 2000 to 2002. And then he was also the one that really helped us to move the Journal of the Society with Springer, the current publisher of our journal. Michael received his BA from the University of Missouri Columbia in 1977, and then in 1980 he went to receive his master's degree. From there he went to the University of Illinois, where he got his PhD in the lab of Dr. Widom, and there he worked on photoautotrophic plant cell culture. He had a long history after his PhD in industry, and like I said, he was the kind of scientist that was always very creative, always willing to try new breakthrough technology. And this is exactly why this session that you're going to see today is supported by this fund. So again, it's a way to remember Michael, and now I'm going to pass the mic to Hurong for the introduction. Thank you.
Thank you, Piero, for the great introduction to Michael Horn. Good morning. Welcome to this session, this year's Michael Horn Emerging Technology Symposium, entitled AI Technological Applications Within Design, Discovery, and In Vitro Implementation. So this should be the slide. My name is Yurong Chen. I'm with Bayer Crop Science. Today, I'm co-hosting this session with Matthew Wellman from Terrena Biosciences, and Nathan Wolfried, Laurier University in Canada. Unfortunately, Nathan wasn't able to join today. Artificial intelligence is changing every aspect of life, but understanding how it can be used to improve our life and help us to achieve our sustainability goals is not something generally understood by society at large. This symposium is inspired by recent advancement of AI-driven biological research. We particularly explore the intersection of AI and in vitro biology, and how to harness the power of AI to unlock and enhance our understanding of complicated biological process and systems, and drive breakthrough innovations in medical research, chemical innovation, chemical and drug discovery, and agricultural product development. So our invited subject matter expert speakers will immerse us in this fascinating world of AI, how AI can revolutionize novel product development, precision medicine, tissue engineering, and three-dimensional organoid design, as well as potential challenges and future directions in implementing AI in biological research. Without further ado, I would like to invite the first speaker of the session, Dr. Christy Taylor, a senior fellow from Bayer Crop Science, give the talk. Her title of talk is Accelerating Product Development by Leveraging Artificial Intelligence in Vitro Biology Research. Christy, the floor is yours.
All right, thank you so much. Thank you for inviting me to give the talk. It's quite an honor to be here today. So first, I'm going to give you a little bit of an introduction to Bayer. Next, I want to give you an overview of the protein AI space, which I'm leveraging every day in my work at Bayer Crop Science. And then I'm going to tell you how we're actually accelerating product development at Bayer by using AI. Next, I'm going to talk about the open source software and the OpenFold consortium that Bayer Crop Science is part of. So I'm putting this in there for legal reasons. Okay. So first, I'm going to give you an introduction to Bayer. Bayer has three different divisions. It has a crop science division, which is used. We do trait development there, seeds and traits, and we also do crop protection as well as some digital farming solutions. We also have a pharmaceuticals division where we're looking at ophthalmology products, cardiology products, and radiology products. And then lastly, there's also a consumer health division, making over-the-counter medications like Flintstone vitamins and also Bayer aspirin. Over the years, we have seen a lot of innovation in agriculture, and Bayer has been one of the leaders in trying to grow more with less and applying these innovations so that we can do agriculture in a sustainable manner. And I'm going to show in my talk how we're leveraging AI now to create products and also generate agricultural products that are sustainable. All right, so everybody has seen these sorts of views of AI. We've all heard about ChatGPT, and we've all seen these images from like OpenAI that are generated that look seemingly real, but are really generated by AI. We know that like the cat with the Napoleon hat with a block of cheese, that can't possibly be real. We've also seen a lot in the news too. In fact, in 2023, there was a call out from the White House really looking at AI and saying how really this is going to help us solve some of the largest problems in society, like climate change. And also thinking about this in terms of applying this to agriculture too, how we can feed a growing world amidst the climate change. So AI can have some very positive impacts for innovation. So what the group does at Bayer, I'm going to focus mainly on the plant biotechnology side, which I'm a part of. And we're leveraging AI to look at products across herbicide tolerance, insect control, and also disease traits for the major row crops for corn, soy, and cotton. And really, when we talk about insect control, we're not just talking about one insect species. We're looking at things across different insect orders, Lepidoptera, Coleoptera, and Hemipteran. Also within our data science group, we're looking at a couple different facets. We have to design things in terms of making sure that these proteins are expressed at the right time in the plant life cycle. We also have to make sure that they're expressed in the right amount and the right location in the plant. And particularly, we need to make them so that they're efficacious. And today in the talk, I'm mainly going to talk about proteins and how we're making them more efficacious, but there's certainly AI being used in the other spaces as well. So now I want to frame this in terms of actual proteins. If we think back to the protein structural determination space, so we've been solving protein structures for about 40 years now. And that's about when the crystallography came about. Since then, we've solved about 180,000 protein structures over 40 years. In addition, there's about 15,000 NMR structures and also cryo-EM structures over the course of 40 years. Of course, the challenge has been to be able to predict structures. For a while, the solve problem, we were able to do this pretty well with small molecules, where we were able to predict a structure of a small molecule from a Lewis structure and have a reasonably accurate model. And this is due to applying quantum mechanics to a small problem or some molecular mechanics. So that's basically a solved problem there. However, being able to predict a protein structure from a primary amino acid sequence has been long the holy grail in the protein science community and hadn't been solved for quite some time. The way that we had been doing this in the past, everybody's probably familiar with this, was where we would have a sequence and we would want to try to model that. So what we would do is we would go to the protein data bank that I just mentioned and we would find a homologous protein structure that had been solved. We would take these sequences, put them together, and then we would generate what we call the homology model. And that was precisely just a model. Eventually, over time, if we were lucky, we would be able to solve the protein structure, and it would be like, wow, that's way different, and these are some parts that are different from what we actually thought. So these models weren't the best in terms of being able to study the proteins as well and to make some fine-grained calculations based on these. So what happened in 2018 was pretty amazing. So DeepMind made the first appearance of AlphaFold in the CAST competition, which is a structure prediction competition. And what they did was they took a primary sequence and they were able to predict 3D structures from that. And you can see the very far bar on the left of the screen with protein quality. The number one was AlphaFold, but it didn't blow away the competition, but it was certainly a large leap forward in the protein structure prediction space. And it was impressive, let's see, but not quite there where it needed to be. So fast forward to 2020, and there were some major revolutions that happened in the protein AI space. And you can see the bar on the very far end, the number one was AlphaFold 2, and it totally blew away the competition. And finally, at this point, this holy grail for protein science finally came to be that we were able to predict protein structure from amino acid sequence. And it really has changed the world. So at first, it seemed like it was, oh, this is a solved problem. We've totally figured it out. However, after studying this some more, there's still a lot of work to be done in this space. We can't just take these AI predictions and use them and count on them for everything. There's still a need for structural biology research to be done. The number of AlphaFold 2 citations has increased fold upon fold, since AlphaFold 2 came out. And the 2024 Nobel Prize was awarded in the protein AI space for the prediction of protein structure, as well as for computational protein design. And so this has really set fire to this field. I want to call your attention to one of the major advancements that led to this, and it's called the transformer. And that is important for being able to focus on particular important aspects. It had been used in the image space. For instance, if we have an image, it's been able to allow focus on certain parts of the picture that are important. For instance, you have a lot of other things in the picture, and for instance, the stop sign. It's able to call attention to the stop sign versus all the other stuff. Well, this is also important in the protein space. It's able to pick up important areas of the protein structure, and that's what's being used to feed the protein folding. Another important aspect is natural language processing, and this has led to new protein folding tools too. So you've seen this before, I'm sure, where you're able to take a sentence, and what you're able to do is mask that sentence. You're able to block out certain words, send it through a language model, and then it's able to predict what words come in that sentence. Well, again, the same thing has been done in protein space. We're able to mask certain amino acids, send them through the deep learning model, and then predict the amino acids then back into the structure. So AI is part of our toolbox that we're using for protein design. You can see that there's data-driven methods all the way down to more first principle methods. And we're really using all of these to design proteins and make new traits. So next I'm going to talk a little bit about how we're using AI to develop new traits at Bayer. So I mentioned at the beginning of my talk that we have solved about 180,000 protein structures. Now we're able to predict lots and lots of protein structures. So there's the AlphaFold database, which has over 2 million protein structures that are folded with AI. In addition, there's the ESM-fold database, which is based on the natural language models that I just talked about. And that database has over 700 million structures. So we almost have 1 billion protein structures to leverage in our design. This is a huge improvement over the 180,000 proteins that are just in the PDB. So it's quite revolutionary. Now, this is only about 10% of the sequences that are in NCBI. So we still have a lot of space to go. But what we're able to do now, given this huge swath of protein data, is we're able to take protein structure now and then essentially do a essential blast against these protein structural databases to find new proteins. And this is exciting because it allows for a lot more sequence diversity, because things can fold to like a similar fold, but be very different in protein sequence homology. So this is one way we're finding new traits. Another exciting innovation are these protein diffusion models. And what this does is it allows us to do generative AI, basically. So I'm going to give you an example in the image space. So we have my dog Lucky here on the screen. And what we can do, they've been doing this in the image space for a while. You take the image, and then you add some Gaussian noise to it, and then you can use an AI algorithm to back out that noise to generate a photo that is, can be different than the photo you started with, kind of doing some hallucination in there as well. Well, what we're able to do now with the new protein AI tools is we can do the same sort of thing with proteins to generate new and novel traits, depending upon how we use the parameters in there. And this is a new thing that we are doing and experimenting with at Bayer. So an important aspect has actually been, there's been an explosion in these protein AI tools since 2020. And I have a lot of different models mentioned here on the slide. Now, these are foundational models. These are trained on a large swath of data that's out in the public. And what we can use is we can use these in terms of like foundational models. So we're able to apply these to many different areas. And a lot of these are open source, so that's super exciting too. So we can apply this to insect control, herbicide tolerance, disease traits, and also we can use this in terms of bioprocess development. We have an arm at Bayer that is looking at making compounds from enzymes for production reasons. It's our biotransformation group too. So all of these things can use these foundational models. And what we can do, especially in the group I'm in, we can use these in these different areas, and that can aid in our design learn cycle. Many of these are tested with in vitro. The in vitro biology is incredibly important in this field because there's no way we can do and accumulate all of this data via in vivo work. So a lot of these, the in vitro biology is really feeding the machine to allow us to obtain data. And what we can do is we can apply some of these foundational models. We can use them without any a priori data in our field, and this is called zero shot. Basically just using the model and making predictions. What we can also do is take some of this in vitro data that we've accumulated, and then we're able to take these foundational models and do what we call fine-tuning. So that's making these models so that we're able to use them for a particular space, for a particular application, and make predictions. So both of these are ways that we're using these models at Bayer. Another area where we've been using AI is development of some enzyme models. So I mentioned we have a biotransformation group at Bayer. And what we do is we're making models for some of the enzyme classes that the biotransformation group is passionate about. And we're also able to then apply this to herbicide trait development. So say we have a herbicide, we're able to, for instance, have a particular reaction that we would want to carry out to deactivate that herbicide. And we're able to use these models to find enzymes to nominate for in vitro studies that would allow that herbicide to potentially be deactivated. Another aspect is leveraging what we now call these multimodal models. That means that it's able to not only predict protein folding, but it's able to predict other things as well. And that's along the lines of what AlphaFold 3 has done recently. It's able to do protein ligand co-folding. So it's able to take the ligand and the protein and actually fold them together. This allows for more conformational flexibility than you would normally see just based on docking. And we can also do protein ligand or protein-protein co-folding too, which allows us to look at protein-protein interactions and structures such as that. So this is incredibly important for the crop science field because in crop science we have so many different insect species and so many different weed species, we can't possibly have crystal structures of all of those. So this is allowing us to do some structural studies without solving crystal structures. Of course, there's still an important space for folks solving those crystal structures. Next, I'm going to talk a little bit about the open source software that we're using and the importance of this. So I mentioned the AlphaFold 2 software and how revolutionary that's been. I'm going to talk now about some of the issues with that. In 2020, AlphaFold 2 won that Cas protein structure competition. 2021, the paper was published. However, we were not able to use AlphaFold 2 commercially. It was only released for non-commercial purposes. At that point, the OpenFold development began because of the need for open source software for this. That put pressure on the DeepMinds group, which then released the AlphaFold 2 license to everyone. However, we realized the need at that point to really generate a consortium for open source software to advance the AI protein space. And that's when the OpenFold Consortium was founded. So the mission of the OpenFold Consortium, which Bayer is a member of, is to develop open source software to drive the protein AI space forward. Because really, we've only scratched the surface of what we can do in the protein AI space. So some of our current projects include the OpenFold 3 development, and that's going to be an open source version of AlphaFold 3, which should be coming out soon. We also have invested some in just some in vitro and biophysical data, because we really think that this is what's going to drive the innovation in this space forward as far as generating more accurate models for us to use. And that's in collaboration with Gabe Rocklin at Northwestern. Also, we're looking to do OpenFold antibody in the future, as well as many other projects. So for industry members that are here, you can scan the QR code if you're interested in potentially joining the OpenFold Consortium. And we have about 20 or 25 industry members that are part of the consortium now. I think I'm going to skip over this one. And really excited about these multimodal models and about OpenFold 3 coming out soon. What this is going to do, like AlphaFold 3, is going to be able to predict protein-ligand interactions, protein-protein interactions, and also protein nucleic acid interactions. So I think this will be another big leap forward in the field. We'll have all the training data out, and it will be completely open. All right, and thank you very much. I really appreciate the opportunity to come to speak with you all today. Thank you.
Nice presentation. I have one question. I don't know how much you can speak to this, but one of the challenges that still exist today in co-folding approaches, especially protein and ligand interaction, is that they are not super well performing as compared to like a free energy perturbation physical mechanisms. I mean, I'm getting excited given what you're coming up with. Can you talk on how much OpenFold 3 is going to be good at these kind of tasks?
Yeah, so you are absolutely right. There are problems with stereochemistry with these protein ligand co-folding programs. It is just the beginning of the development. And one of the cool things about the OpenFold Consortium is it's part of the Open Molecular Software Foundation, which has other components like open force field and open free energy. So one of the things we're looking at is potentially trying to put these together, right? Because it's going to take innovations in both fields. So it's going to be like an AI-driven, but then also potentially having some cleanup maybe with the open force field consortium so that we're able to make sure that our bond lengths and distances are proper, maybe also combining with MD. Because you are absolutely right, the AI isn't quite there to be able to completely model it in the way that comp chemists would like. So we're really trying to drive that forward into a way that industry can use the software the way that we strive for.
Good. Excellent presentation. I would like to hear your thoughts more about the regulatory perspective of these new proteins that they don't have. Obviously, they don't come from nature. They don't have a history of safety use. And also about what it will be the public perception of these more synthetic, unnatural proteins.
Yeah, so that is something we're currently working with our regulatory organization on. In addition, we have the sourcing of species that we typically use when we go and have our products go through regulatory. I am not sure how it's going to fare in the future. We are going to make sure that we are following all regulatory aspects, before we would actually put them in plants. So we're going through step by step and seeking the help of our regulatory colleagues at Bayer. It's outside of my area, but I think it's something that the field definitely needs to think about and make sure that we're ready to do, because the protein AI space is certainly moving in this direction, and it's moving in this direction for pharmaceuticals for sure. But it will take some regulatory hurdles to get there.
I think we'll move on. We have another 10 minutes at the end for the questions, and also if we have more questions, catch Christy at the coffee break. I think the next talk will be delivered jointly by Kelly Chapman and Suzanne Pose from Viscon. And their talk title is AI and Automation Implementation, Challenges, Changes, and Successes. Kelly, floor is yours.
Thank you very much. Well, good morning, and first we'd like to thank you for inviting us to be a participant and a speaker. Viscon would like to share our view on AI and automation implementation for tissue culture and as it was said, the challenges, changes, and successes. And today, I will be joined by one of my colleagues as a presenter, but Viscon is based in the Netherlands. The company has been automating processes across multiple industries for more than 50 years. We're constantly looking for ways to push the envelope and to automate challenging processes. I am the North American representative for Plant Technologies, and as I said, the delivery of this presentation will also include our research team lead, Suzanne Poles, who is here from the Netherlands. To simplify what terms we are talking about today, I want to just give a very simplified definition. If there are any experts on AI and automation in the audience, I'm sure there are lots of technical aspects we could argue about this. But the point of this is for us all to be on one page with a simple definition. Using computer models and learning to build machine capabilities independent of human interface is what we are going to consider artificial intelligence for this talk. It always must be trained in the beginning by humans who do have that information and knowledge. And automation we will think about as being the use of mechanically driven actions to complete a previously manual task. This may or may not involve one or more of the following, which could be robotics, computer vision technology, software, computer modeling, machine learning, or artificial intelligence. And I'd like to take a step back for just a moment, because quite often we get caught up in the most recent and high-tech technology, but I want to remind you that automation could be as simple as a motion sensor turning on a faucet when you go to wash your hands. So there are ways to begin automation in simple steps. It doesn't have to be a high-tech, expensive machine when we talk about automation. Automation requires the intersection of multiple disciplines and activities. You have an organism. Here, of course, in this group, we're talking about both plant and animal-based cells, organs, or organismal levels. We also have a machine of choice, probably includes traditional mechanics, includes robotics. At this point in time, that's very popular. Vision technology, software, machine learning, and artificial intelligence. And then the third leg of the stool here is that we have people. And these may be people that are operating the machine or people that are currently managing the manual processes. They may be auxiliary people involved, but all have to be involved in the process initially of automating an activity to make it successful. All of these come together to result in your standard operating procedures and processes. And that, as we will talk about more in this presentation, is extremely important. So if you're thinking about whether or not to automate, then why are you thinking about automation? Hopefully not just to be popular or in the most current trend, but before you start that process of automation, you have to have some clear objectives. Your objectives may not be the same as another lab that's beginning the automation process. Are you looking for something that it frees up your personnel? Maybe it decreases mundane tasks or improves consistency and quality. Maybe you're bridging a labor shortfall during a high production peak. You have to document your current status and your target goals in order to be successful in the automation process. And so what are your options? We're all here because this is an in vitro biology conference, and so, for instance, we have in the past automated many of these processes, or all of the processes actually listed here, have been successfully implemented. And they can be anything from simple media filling into a container to more integral processes of data integration. It could be actually transfer of tissues by automation. And for one example, here in the United States, we don't see automation quite as much as we see in the Netherlands where Viscon is based. So for more than 20 years, Viscon has been able to, on a production scale, actually grade and sort very tiny plants so that people had a consistent quality within, let's say, one tray of plants. So if you didn't have consistent germination, we have machines that can then take that tray, look at each of the plants, and then sort them into trays by size. Really, this is phenotyping to most of us. And so we're transferring that type of knowledge from what has been production systems into now R&D and breeding systems that we are very excited about. You know, you can automate anything if you have unlimited funding and unlimited time, but typically that's not where any of us are. So people usually are looking for automation that makes sense. You need to assess the cost and the return on investment. And it's really critical to know your processes, to be able to document those, to be able to communicate those. And then you have to determine if your team is willing to flex or to make changes or adjustments that may make automation more realistic for your processes. You have to involve other teams in your facility. That may be your maintenance team. It may be your client, so to speak, that you provide materials to. It could be people from your support systems. And you have to be realistic on timelines. It takes time to document processes and to scope out what the best fit for your automation may be. And at this time, I would like to turn this over to my colleague, Suzanne Poles.
I think I dreamt around 4 times last night how I totally, fell on my face there. Luckily that didn't happen. But thanks to Kelly for transitioning here. So this is just the moment where I'm stepping in and I'm going to be explaining to you guys a little bit the process we have at Viscon when we start considering automation and how we would innovate on different steps within your process. And that's really something I'd like to emphasize at this moment is, as you can see here, this is not the pointer. So it's really about your process. We all have a different setup and we all have different protocols, right? And this can have small changes depending on how your wet kitchen, your plant handling, or even your growth chambers look. And it's key for us to be able to distinguish between the consistencies and also the differences in those processes, and what would be key for you to be able to automate versus what are standard functions. Usually, this process would start with a mapping. We sit with you during pre-consultancy and we have a little bit of a discussion around what does your process look like and how would you go from A to B. During that process, we also identify certain functions. And as biologists, we're very used to working in certain protocols that are standardized. But working closely with the engineers in our team, I've learned that during those process mapping stages, it's also really important for us to understand what type of functions are we identifying. And that could be something like transferring something from A to B. As you can see when I go forward, that you have just simply putting a ball in a box and moving that box forward. Considering that process, would it not be possible if you just moved the 2 boxes closer to each other when you're transferring that ball from box A to B? This takes a little bit of a moment for you to consider, why have I always placed the boxes so far from each other? And what would be the advantage in being open to changing that process slightly? What I often compare this to is like having a bunch of 3-year-olds with me in my lab. They're constantly, when I'm working with my engineers in trying to optimize these processes for you, they're constantly asking me that why. Why are you doing this? Why have you always placed it here? And sometimes those are pretty annoying questions because we simply say, well, it's always been done this way. My professor taught me to do this way. I was told that this is the best way of doing it. But if you take a step back and you really consider, the fundamentals behind why you're doing it, there could be a simple change in making it more efficient. And in a simple way, that it's just the same way as how it goes for what we're choosing to automate, right? There are certain limitations to what the mechanics and the software is able to do at the moment. For example, you have a tissue culture lab, and we function very often in the plant sphere, as Kelly has mentioned earlier and pointed out, and that's where the examples are going to be focused on for me today. But when you're considering a tissue culture production lab or even an R&D lab, and you're going through your multiplication process, you have those lab technicians working in the laminar flow hoods, and there's a certain level of dexterity that your hand is, for example, very apt at. We were designed amazingly. A robot, despite the amazing technologies we have at the moment, does not have the same dexterity. It has limitations. And when you are wanting to mimic that process in your lab, unfortunately, we have some limitations. And we have to work closely with the engineers to understand what limitations there are towards the dexterity that those robot arms have, and to what extent we could mimic your process. Another one is the great developments in artificial intelligence, which, as Kelly has also pointed out, is a term that's very often and very broadly used at the moment. And in the previous talk, it was also pointed out that AI is amazing, but it has its limitations. We're not at the point yet where that intuition, that level of troubleshooting, we are all adherently taught during our pre-grad days and as scientists, that does not necessarily translate directly to AI. If you're working in a lab and suddenly you realize you have a different level of agar concentration, and you need to adjust the level of force you're using to put your colors into that new container, the robot does not do that automatically unless we have taught it to do that. And thus, there are certain limitations to the variability that these technologies can deal with. And that is something that you really have to consider when you are looking into automating your process. I work very closely with engineers that think a little bit differently than I do. And when I started at Viscon a few years ago, I remember very starkly sitting in a room with a team of 10 or 15 engineers, and they were looking at me and saying, Suzanne, these plants are quite clumpy and very bushy. This doesn't really work very well when we want to automate this process. And one of them looked at me and said, well, can't we just stretch the plants? I mean, that should be easy. And at that moment, I'd only been working a few days at Viscon, and I remember my brain going to those very archaic torture systems that you have in France, where you're tying the people at the 4 different limbs, and you're literally pulling them apart, right? And I just thought to myself, no. And I remember looking at him going, no. And I did not in that moment, for example, immediately think of, okay, looking at different light spectra, trying different, hormones. I was just really envisioning this torturing my little plant and going like, no. And in that moment, I realized a very important key in making automation a success is communication, right? It starts with how you're able to communicate within the team, how you're able to translate your needs, your process, and be able to put that into a language that is universal for everyone in that process. As I mentioned, we work specifically with plants, mostly in our automation and with my team, as I'm a plant scientist by trade. But the thing is, you also have to be able to translate the biology, which is super basic to you and something that goes without saying, towards someone that does not deal with as much variability as you do every day. Nuts and bolts come pretty standard. Robots have certain axes that they move in. There isn't a lot of room for variability in the eyes of the engineers. Oh, and don't get me started on the software people. They have a completely different way in the way that they approach what logic is. And I was sitting there and thinking to myself, oh, heaven help me. So being able to integrate through successful communication your 3 disciplines is also really key. And I think probably the most basic part, which was also mentioned yesterday, a lot of patience and planning. We'd all love AlphaFold to have been existing 20 years ago when we were doing our PhDs and you had to do those crystalline structures and your protein extractions yourself. It didn't. It took time. And therefore, our automation also sometimes takes a lot of specific planning and development. So I'm going to take an example of a project that I was very closely involved in. This is a very specific example to our company, where we, 2 years ago, had the privilege of launching a machine that is functional, or a robot that is functional within the multiplication phase that you have of organogenesis or meristematic tissue multiplication, right? So this is basically where you would often have a bunch of people in laminar flows. Their basic job is just chopping up your plants, making a bunch of baby plants called explants, and putting them into fresh media. And we looked at that process and everybody was saying, well, it would be great if you guys could automate that chopping up of the plants. And we went like, yes, it would be really great, but that's probably not going to happen in the next 5 years. And instead of just saying, okay, well, we can't help you, we said, which part of it is really easy to automate? Which function would be possible at the moment? And one of those functions were simply picking up those newly created explants and placing them in fresh agar. That sounds pretty simple, right? How hard can it be? You have to see the plant, you have to pick it up, you have to stick it in a new cup. That didn't sound very complicated. However, when we started with this development process, that's really when I started to see that communication can be extremely key at how you mitigate the variability you have to deal with every day. One of those variabilities would be biological variability. So when I started at Viscon, I had to give a lot of presentations about what on earth is plant tissue culture, why would you use it, what are the advantages, and one of the key terms that we would mention was that plants are clones of each other. Yeah, that did not mean the same to me as it did to the engineer. They thought every plant literally looked exactly the same. So I had the engineer coming to me and showing me this photo that you see on your right. And they were like, plants don't look the same. And I really had to take a moment and step back and go like, well, duh, of course they don't all look the same. Do we all look this? Why are you asking me this? But to them, they really envisioned that each of those plants would look exactly the same. The second challenge that we had was not only biological variability, but also human variability, where when we started to define that process in everybody's lab, we realized that it's maybe not as consistent or structured as you anticipate. Not everybody makes their stock solutions the same. Not everybody adds their hormones the same. Not everybody weighs on exactly the same scale. And those tiny pieces of variability, that really had a large effect on how successful our automation was. And then, not only being able to identify that variability, but also being able to deal with that variability. Is it necessary? Is this something that my machine needs to be able to cope with? Or is it just something that you simply have to try to standardize within your system? And lastly, if it is something that is really important for your system to deal with, well, how do we train it to deal with that variability? And a simple example would be, where do the technicians choose to pick those explants as to not damage the meristems? Because that was a key thing we kept on telling the engineers. You cannot damage that growth point. You have to give a certain level of force. Don't pinch it. Be careful. And then they came to us with pictures we took. These are some strawberry explants. And they were like, okay, Suzanne, if you had to choose where to pick it, if you guys were looking at these blue lines here, where would you choose to pick it up? And I gave my annotation. Funnily enough, we're a few different researchers in our team, and when the next researcher had to identify where she would pick a plant, that was literally millimeters away from each other. And they came back to us and they asked us, but why? why would you pick at a different place? And I don't know how many of you guys have actually considered this. Why are you pipetting like that? Why are you holding your tube in a certain way? Why do you choose to use that tube? Well, I haven't think about it that much in that much detail at that point. Now, unfortunately, I have, and it gives me nightmares sometimes. The third and the last variable point that I want to emphasize is technical variability. We don't all use the same plastics. And plastics was actually one of our largest challenges with this development. For the different, they call it product carriers. The engineers, we call it just simply cultivation cups or consumables. One of the biggest thing was defining the different sizes you have within your lab, because that was really key for them. If we wanted to automatically open or close those lids, how difficult would it be for an Eppendorf tube versus a Falcon tube versus just a normal cultivation cup that's often used versus a Petri dish? You have different shapes, but you also have different rigidity in plastics. I don't know how many of you guys are polymer scientists and have studied plastics, but that could be quite a complex system if you have to start to understand what different types of plastics you have inside your lab and how to mitigate that. Another simple example was this machine is really key at sticking those explants into the agar, right? But we don't all use the same level of agar. So your rigidity of your growth media differs. How much force do you need to use to not squish your plant? And that was something that I really, emphasized a lot to those engineers. If you squish my plants, I will come and find you and haunt you until you die. So they were all like, okay, Suzanne, we're not going to kill your plant, in the end. However, that was really something that was key in the end for them to understand how to program that robot. Going through all that pain, and going through all of those different challenges, we were really happy to say that once we started implementing this automation within different lab processes, we saw that there were also a lot of unexpected joys of implementing these new technologies. One of them was process improvement and continuity. You now do pick every plant at the right point. You do use exactly the same force between every explant being created. Also, the robot doesn't get sick, right? It can work 16 hours, right? You don't work with a carcinogenic and die at 40 due to the ethidium bromide in your PCR or your gels you had to run. And then I think the most important that we see that is really also key is, of course, the economics. There's a lot of cost decrease when you start to really analyze your process and not necessarily always have to implement the automation, but also have an increased efficiency in your process. So that was just an example of one of the innovations we're working on. I think most of you guys know we also have a lot of other exciting ones we're working on, but to close off this, Kelly will take it further. So we have a bit of a tag team we're doing.
Thanks, Suzanne, and to all of you for your attention. And we'll try to finish up quickly here. So basically, there are many successful examples of automation, and everything listed on this slide, as I said, has been successfully completed or is near commercial completion. We did not include a lot of these things in our presentation to avoid it being commercial. We obviously would be more than happy to give you more information on any of these. Just very quickly, I would like to say that typically we do go over the business case whenever we work with clients, and that's ultimately important for you to have the right information to put into those types of calculations. With much of these machines, there is a less than 2-year return on investment. Also, in terms of the future, we feel like integration is one of the major topics, whether you're talking about integration of different processes and automation across all those processes, or integration of software solutions, integration of these different technologies. So integration as a whole is key to the future of automation and to any of you considering such. And The new challenges, we believe, create new opportunities. VisCon has a tradition of trying to solve problems that other people really aren't typically even tackling in the industry. And we've already talked about that includes plant science or biology, engineering, computer science, but most of all, it really comes down to a good collaboration between the user and the provider. So whether you work with our company or another company, that communication and collaboration is definitely key. And again, I will skip over this, but some of the new technology has been talked about in both the talks so far and will continue to be developed. And we hope that in the question and answer session that maybe you can give us some of your ideas about what is your current greatest need for automation, because we're always trying to glean information and ideas from industry and from academia on what do you need and what would be of benefit in terms of new technology. So we are working on automation for production and R&D and breeding, and we would welcome you to stop by our booth, and our emails are here as well. Thank you very much for your time and your attention.
So we'll take time for questions for this one at the very end. So our third speaker is Dr. Stefan Birkenhoff, where he's currently an associate professor in UVA's Biochemistry and Molecular Genetics Department and director of bioinformatics of the Precision Immunomedicine Initiative, working on development of classical and quantum machine learning approaches for biomedical applications. Please welcome Dr. Birkenhoff.
Thank you. And if it's approved by the board, I'll be promoted to full professor. So that's kind of fun. All right. So thank you, organizers, for inviting me. This is an exciting opportunity to present our work, and you'll see it's a big team. So today I'm going to be talking about a precision medicine, immunomedicine initiative that was funded at UVA. This is Colleen McNamara, a physician scientist and a cardiologist, Angela Taylor, a clinician scientist, and me wrote a grant, and it was funded to start a program, and I'll describe that in a little bit. I'll give an example of a pilot study we performed, and then I'm going to go into an explainable AI framework that me and a computer scientist, Adong Zhang, and a student developed for single-cell RNA-seq, but we applied to this pilot study, so I'll show you those results. And then going beyond a pilot study, I'll describe a cohort that's much larger that we're working on. And then finally, I think you all know single cell is pretty expensive. So we actually developed an AI framework that takes bulk RNA-seq and generates representative single-cell RNA-seq data. All right, so cardiovascular disease is the #1 killer in the United States and worldwide. And you can just make that as an unqualified statement. And increasingly, we're understanding cardiovascular disease and atherosclerosis as a chronic inflammatory disease. And here I'm just highlighting a review where the factors that maybe mute atherogenesis or that promote it, the blue are the good guys, the red are the bad guys, are playing a role and increasingly being understood mechanistically. And my colleague, Colleen McNamara, is an expert and focuses on B1 cells. They produce IgM antibodies that actually bind oxidized lipids and are protective, it turns out, for atherosclerosis. And here we also show some cytokines. So as I said, in response to this, we got an initiative funded that at its heart is focused on studying the role of the immune system in cardiovascular disease. This involves multiple schools in the university, and at its core, what we do is patients that are indicated to receive advanced imaging, which includes intravascular ultrasound or computed tomography, angiography. These are advanced imaging modalities that assess your risk of cardiovascular disease. So these patients receive this, and when consented, we draw their blood and we carefully cryopreserve it for further experiments, especially single-cell proteomics, either mass cytometry or multispectral flow. We also preserve their DNA. We extract their DNA for genomic studies, and we can do other omic studies as needed or of interest. We bring that together and perform integrative analysis as well as AI, and I'm a computational biology. I lead the computational biology group. So what's an example of a pilot study? So we have, I'm going to forget the controls. For our pilot study, we had 16 heart failure patients, and we were blinded. Some received anakinra, and others received placebo. But after 6 weeks, we assessed whether or not they improved. And so the goal of this study ultimately was to identify patients that we could predict would improve. And this is important because when you give something like anakinra, IL-1 receptor antagonist, that can lead to serious side effects, including, infectious disease. You become more vulnerable. So that was the goal of the study, and you can, in the lower right, you can kind of see how the VO2 study, a cartoon of how the VO study is executed. So what was the study design? So at week 0, that's baseline. The controls and the heart failure patients, we got their blood at that time, and then after the 6-week period. And then we stimulate the cells with vehicle IL-1 beta. We extract their PBMCs, I should say. And then we stimulate them with vehicle IL-1 beta or IL-6. And this is actually an in vitro model of what the cytokine environment might be. And what I'll show you, we actually measure functional markers, the functional state of these immune cells, including, like, for example, you'll see phosphorylated transcription factors like NF-kappa B and the stats. So, and then we take it through mass cytometry, which basically, if you're not familiar with it, is just single-cell proteomics, okay? So I'm just going to jump right to kind of the kinds of analysis we perform. So this is the, kind of the single-cell analytics that go into analyzing mass cytometry data. And so in the upper, and right now I'm focusing on T cells. So in the upper left, I'm showing you a UMAP representation of the different colors are just different cell subtypes of T cells. In the right, you see kind of along the x-axis, if you will, type markers. These are markers that identify the different cell subtypes. You can see the colors on the far left of that heat map. Those are the cell types that correspond to the colors in the UMAP. And just the high and low markers of particular cell subtypes identify them. So you can, on the right, we annotate them. We put names to them that are in the literature. And then in the lower panels, you see kind of measured levels of, in Th17 cells, phosphostats, phospho-NF-kappa B, phospho-ERK, and P38. And these are for patients at baseline. These were patients that were at baseline and improved. Baseline didn't improve. So from that, we can identify and see if there are markers that identify patients that will or will not improve. We're particularly interested at baseline. But I'm not going to go into the details of the analysis. I'm just going to point out that based on the literature and some preliminary experiments that Colleen and a postdoc performed, they honed in on Th17 cells as possibly being the interesting cells to focus on. And I'll just come back to that in a second. All right. So, so where does AI come in? So as I'd mentioned maybe in the very brief outline, we developed a single-cell explainable AI framework that works in the following manner. It basically, you can see there's a cell embedding module where we have what's known as an autoencoder. That's on the left. It just basically learns the representation of the single cell data. But it does so that in the middle, it clusters the data and identifies prototypes for every single cluster. So you can think of those as like centroids, if anybody's familiar with k-means. It just identifies these centroids, and from that, that's like at the very core middle identity of that cell type. And then any cell in that cell type that's its distance from that core kind of defines how much it's a core cell type of that kind. That gets coupled to the top classifier, which again reads in individual single cell profiles. And each cell gets a vote, both of the class that you're trying to predict. In the case of the Red Heart study, it's whether patients improved or not. So the input is the single cell data, the output is the class, improved or not. And every cell gets a vote. In fact, it votes for both, but it weights it. If the cell is voting for improved, it'll give a very high score for improved and a low score for not improved, for example. And that gets coupled to the degree it's at the core identity. In this way, each cell gets a vote. We aggregate all these cells' predictions to make a final prediction, but we're also able to identify which cells are driving the predictions. In that sense, it's inherently explainable. So let me take you through kind of what the results look like. First, I'll describe how we implemented in the results of this tiny pilot study. And I should say, here, you know, these is again, single cell is very expensive, and you usually work with very limited patient numbers. But the other thing we learned was this framework is powerful because lots of cells are generated with single cell per patient sample, so there's lots of information. So in one sense, the models just get lots of data to learn, and that's what AI loves. and it just turns out that we get high prediction accuracy, even in cases where it's ridiculously small numbers of patients. So for this Red Heart pilot study, we used the baseline data. Our goal was to predict whether patients improved or didn't improve after 6 weeks. Our training data had nearly 1 million cells. This is one of the things that gives us power from 8 patients. We used 4 patients for what's known as validation, but we really didn't do anything, so it almost served as holdout test data. The formal holdout test data was 4 patients. And on one particular, the best split, we achieved 75% accuracy and 87.5% accuracy when you include the validation data. So what do the explanations look like? Okay, so here's the training data, and for each one of these plots, at the very top, I'm showing you the status, whether the patient was truly improved or not. And then on the x-axis, what you see is the score. Every cell gets its score for improved or not improved, okay? And let's focus in the lower right on patient 1064. You can see that for improved is low with the blazing red, but it's relatively high for not improved. And so that's the majority of cells are voting very strongly for not improved. That patient we were able to train as not improved quite easily. If you focus again on the bottom at patient 1034, that patient was improved, and if you look, the blazing red is very high for improved, but low for not improved. So that was a patient that was relatively easy to train. The cells are voting in the right direction. But now look at patient 1096. You can see both the scores for improved and not improved are pretty strong. That patient was probably hard to learn. And so for the validation and test data, here are the same plots, and you can see we only did not predict one patient, 1007. We predicted that patient incorrectly. Everyone else we improved correctly. And if you just kind of look, you can see where the blazing red is and the label at the top, and they kind of correspond. So that's the performance on this tiny data set. And one of the computational biologists in my group developed a movie during training. So the models start with random weights, as is common in machine learning, and so they don't have an opinion. And they form an opinion as you learn. And so this is just a nice way to possibly diagnose these more difficult cases that we can't predict. And then we can impose these movies and their scores on the original UMaps of the cell subtypes. And so on the right, you can see kind of let's call it the trainer and the dog. And you can see we're now imposing essentially the difference between each cell's not improved minus its improved score, and red corresponds to improve, and blue corresponds to not improved, to improved rather. So red, I'm sorry, is not improved. Blue corresponds more to improved. And so at the end of training, here's just some of the results. So I'll remind you in the very bottom, these are the T cell subtypes that we clustered and annotated. Okay, those are just the cell subtypes on a UMAP projection. If you focus on the red on the dog's head, that's cell type 1. Those are Th17 cells. Now, for vehicle, IL-6 and IL-1 beta, you can kind of see there's not much of a difference, at least at this stage. So you can focus on any one of them. They look about the same. And look at the dog's head. You can see that it's blazing red. And this is, again, this difference between not improved and improved. And you can see the Th17 cells are identified. So even if we never talked to Colleen about Th17 cells, the model found the Th17 cells on its own. And Th17 cells from the literature are associated with heart failure when they're dysregulated. So this was a pilot study. We've now done a study with 214 patients where they received intravascular ultrasound. And so from that, you can quantify a lot of measures of cardiovascular disease risk, including atheroma burden, percent necrotic core, the calcium, whether the tissue is fibrous or fatty. You don't want these scores to be high. And so because they're measures of cardiovascular disease risk. But what's really important with this is, again, we have 214 patients this time, and we have 700 million cells. So these are the numbers that AI likes. When you think about language models, they actually train on trillions of tokens. And it's known that scale made a huge difference. We're actually working on LLMs too. I'm not going to talk about that. But scale makes a difference. And so we're very excited by this data set. And here I just show the breakdown in terms of millions of cells of the B cells, the T cells, the myeloid cells, and the NK cells. So, and here's the panel that we use. This is a spectral flow, and you can see the markers that we use to identify B cells, T cells, monocytes, dendritic cells, and NK cells. We cluster them grossly first, like to separate the B cells, the T cells, the monocytes, the dendritic cells, and the NKs, and then we subcluster those, which I'll just show you the NKs as an example. But we also have molecules that assess immune checkpoints, the activation state of the cells, as well as chemokine receptors, and of course we check for viability. So I just want to point out one thing. If you do analysis, so your conventional R-based pipelines, say, or even Python to cluster, like do single-cell analytics, will not work on data sets of this size because if you look at the CPU, if you ran CPU and you try to perform what's known as Leiden clustering, that's network-based clustering, even on 20 million cells, it would take about 400 hours, and you have to do it repeatedly. So that's a non-starter. So we identified tools that are set to do clustering on GPUs, and that really does the job. So I just want to let anybody know who's doing this stuff, because we are going to everybody's going to be getting more and more cells as the technology gets better. And maybe at some point, the GPU-based approaches will be incorporated into conventional pipelines, but just be aware of that. So here now we can do, for example, nearly 85 million cells in 6 or 7 minutes. And so that's really fantastic. And on the left, we're showing you our NK clustering. And kind of, even with the NK cells alone, you see on the right, the bigger picture, the gross NK clustering. And when we still see type markers that are bimodal in their distribution, that means that there could be more separation. And our biological annotators say, yeah, I could see more cell subtypes. We further cluster those. We take those apart and we further cluster them. But we do multiple rounds of clustering, so you really need an efficient approach. So we are still working on this data set, and we're soon probably going to start doing the AI and the NK cells first because it's the furthest ahead in annotation. Now, I keep saying that single cell is expensive, and it is. It still is not sufficiently cheap. So we developed a framework called bulk to SC that if you have single cell data from a particular tissue, you can and you have bulk RNA-seq data. If you have single cell from a particular tissue and you have lots of bulk from that same tissue, you can deconvolute the bulk in principle. And so I'm going to show you how we do that. So again, we use an autoencoder where we learn the same representation of the single cell data as output, but we do so in such a way that in the middle, we learn the distribution, the Gaussian distribution information for every cell subtype. And all we need to induce that information in the middle is what that cell subtype is. That allows us, if you focus on the lower left, to use any deconvolution tool that takes bulk RNA-seq data. There are lots of them out there. There's CyberSortX, there's TAPE, there's Dissect. I could keep going on. There's a Bayes Prism. But we use TAPE. It worked well in our hands. So what that does is that takes the bulk and it predicts the proportions. And these frameworks are training on single cell data as well. You can sample it and you know ground truth and you can just build these models. And they're high performing now. So again, in the lower left, you take bulk RNA-seq data and the output is the cell type proportions. Now we use that as the front end of our framework, and then we use the back end of the thing on the top, going from the middle to the right. And so the way it works is you feed in the single cell data, it predicts the proportions. From that, you can sample what fraction of cells you need for every cell subtype. All you need is the label to induce the distributional information in the middle of that cell subtype, and then that generates the representative single cell data on the output. So how does it do? Okay, so here's a single cell pancreas data set that we started with. We use 75% to train, and then we use 25% on holdout data. The left is the original data. The right is generated from the holdout pseudobulk. It's just generated. And by eye, you can kind of see it looks pretty good. And we perform a number of quantitative measures to assess how well it's doing. So on the left, once again, is a UMAP representation of the original and the generated in the same embedding. So you can see how well they're peppered. And the more they're mixed together, the better. And you can see, you can kind of hardly tell them apart. And then in the right two panels, in the original, these are 150 of the most variable genes, and these are gene-gene correlation plots. And you can see that the original and the generated form very close to the same gene-gene correlation plots, which is a pretty tough measure to achieve well on. And we even flatten this and calculate the correlation coefficients. And for a lot of our data, they go between 0.8 and 0.9 of the correlation of correlation, so it's pretty high. But we measure how well it's doing even further. So one of the colors was bleached out. I can see that now. But so there would be two colors in the left panel for original and generated. But you can kind of see that the distributions are kind of on top of each other anyways. And so what the cell detection fraction is for each cell, you quantify the fraction of genes that have non-zero values. And so we get the same distribution essentially between the original and the generated. And there's another measure that you can measure of local mixing. So basically what it is called a Lisi score. So locally, if, let's say, you're original cell, and you're just surrounded by original cells, or you're a generated cell and you're just surrounded by generated cells, your Lisi score would be 1. But now let's say you're beautifully mixed. Generated and original are just beautifully mixed. Your Lisi score is 2. And that's what you want to see. And if you look at the UMAP representation of Lisi scores, you want them to be more toward yellow and green, which they are in our case. And then we also just have since it's a local measure, you can calculate a histogram of Lisi scores. And from the point of view of the generated data, and from the point of view of the original, and the distributions are pretty much on top of each other. And this was not even our best performing data set. Our best performing data set was this PBMC68K data set. And once again, you see the UMAP, they're all kind of peppered in together. The Lisi score UMAP looks fantastic on this one. And then here you can see the original gene-gene correlation and generated gene-gene correlation. And they're not exactly the same, but you can see a lot of the nuances are captured in this. So we're pretty excited by this framework. And we're now working with a colleague. I'm part of a Leduc collaborative network. And so someone at Mayo I'm working with, we're working on, they have single cell atherosclerotic lesion single cell data from mouse, and then but they have a lot of bulk, and they were caught in this issue where they didn't want to generate the single cell for everything, but they would love to see what the data would look like. And that even allows you to maybe pick a particular sample that's the most impactful from the prediction, and then do that experiment, and then that can maybe what goes into the publication, but you can pick a high bang for the buck single cell sample. So with that, I'm going to end. There are plenty of people to acknowledge. This is really a big team effort, but in particular, I want to acknowledge Colleen, my colleague, and Angela Taylor. We started this iPrime initiative, and then Miles, Kim, and Corey Williams, who did a lot of this analysis. Thank you.
We have time for one question.
Hi, thank you for the amazing presentation. The bulk to single cell tool is really interesting, and using the deconvolution. I just have a quick question about the sample you are using, the data you are using actually. So for the training and test data, you use the single cell and the bulk RNA sequencing data from the same piece of tissue or from the different mice or different human sample?
It's the same. And so what we wanted to do, so what I'm not showing is that we compared to other methods. And as a start, we wanted to know the ground truth, the actual ground truth. So we actually take 25% of the original single cell, we turn it into pseudobulk, but the model's never trained on it. So that we know comes from the same distribution in the single cell. So when we overlay them, we know they're on top. We are now beginning to actually work with tissue that's the same tissue and then actual bulk. The challenge is, as you may know, you can get differences now technically in the way the single cell was generated and the bulk. Single cell tends to be 3' biased, bulk tends to, you actually get the full transcripts well represented because you have random primers that, so that's a difference in the way, one technical difference. There are other unfortunate differences in the actual cell types, cell proportions captured in the single cell. So that takes data normalization. So what we wanted to do was just test ourselves on ground truth, and we compared to two other methods that they're not dedicated to this task of generating representative single cell from bulk. They actually are broader frameworks that do that in the middle. So there's one tool called bulk to space that is very ambitious that tries to generate spatial transcriptomics data from bulk. And there's another method that we compared to called SC Semi-Profiler, which actually what they do is they're designed for large cohort studies. And what they do is their best kind of data is when, let's say you have 100 patients. You have bulk for everybody, but you have single cell on 5. They try to choose the best samples to actually spend your money, and they actually talk about cost throughout their paper, to do the single cell for, that's bang for the buck, and then the rest they generate. And so we compared to those two tools. We compare very favorably, but we wanted to use ground truth for that reason.
Thank you so much.
Thank you.
Our final speaker today is Dr. Zen Ma, who's an associate professor at the Department of Biomedical and Chemical Engineering and a member of the Bio-Inspired Institute at Syracuse University. Dr. Ma's lab focuses on human-induced pluripotent stem cell technology for cardiac mechanobiology, organoid engineering, and extracellular vesicle therapeutics. Please welcome Dr. Ma.
I would thank you so much for the organizer to invite me to this wonderful conference. This is my first time being here, and I learned a lot from all these colleagues. So today I'm going to talk about how you can use more simplified AI models specifically targeted to your own experiments. In our case, it's cardiac organoids were generated from pluripotent stem cells. Today, people has been using stem cell technology to grow different type of tissue in the lab. The basic concept is just using pluripotent stem cells, growing a special matrix, induce the differentiations, and from there, you can generate whatever the tissue that you already have in the bodies. In the field, people has been generate organoids in this like three-dimensional tissues mimicking different physiological function tissues and in our bodies. And particularly in our lab, we're focused on the heart. Just talk about the heart. People has been generated the cardiac organoids, we also call cardioloid, using different approaches. Most approaches is using these three-dimensional aggregates of pluripotent stem cells in suspension culture or matricial embandings, and induce the cardiac lineage differentiations. And from there, you can see the cardioloid have all the key cell types that you can see from the heart. And more importantly, you can also see this nascent chamber formation that can represent the early development of the heart chamber in our bodies. As a bioengineer, we have focused on how we can improve the process, how we can generate a cardioloid with better functions, and more importantly, better reproducibilities. To do that, people has been developed a lot of effort to generate a cardioloid using three-dimensional hydrogels as a replacement of the matrigels. People has using microfabrication techniques to create this micropattern cardioloid with confined geometrical shapes. And people also using microfluidic device to create organoid in a organ-on-a-chip systems. And using these more reproducible and highly functional organoid systems, people can study the different diseases using patient-derived iPSCs or CRISPR-engineered iPSCs with specific mutations. Or people can study how the organoids develop in the lab to do mimicking how the real organ development in the baby. And from there, people using this organoid system to predict the drug toxicities to specific tissue or organs. And today's talk, we primarily focus on techniques that we have been using in the lab for a decade. It's just microfabrication techniques to generate more reproducible cardioloid. So I'll probably just simplify the slides because this has been published a couple years ago. But what we can do is create this couple hundred microns macro patterns on a 6-well tissue culture polystyrenes in order to control the development of the cardioloid. So this is what you can see. So we can create that like a macro pattern system, use a packed hydrogels, and from there we can create it like a controlled colony of the human-induced pluripotent stem cells. What I show here is just a circular patterns with different diameters from 200 micron size in diameters all the way to the 1,000 microns in diameters. After micropatterning process, the pluripotent stem cell can maintain their pluripotencies highlighted by the key pluripotent markers, NANOG, OCT4, and SOX2. And after we micropattern the iPSCs, what we do is just induce the cardiac differentiations to generate a cardioloid in this micropattern substrate. So, and by day 20, we will do all the function analysis for our cardioloid. So this is what you can get by all the differentiation protocols. You can see these are three-dimensional cardioloid systems. The red color is the muscle. Green color is supporting layers on the bottom of the organoids. We just stack this cardioloid on the tissue culture polystyrene. You can see the bottom is a perfect circle because we micropatterned the organoids. And of course, you can see there's a lot of cells only show the nuclear stainings, but now they are either stromal cells or the muscle cells. And we can generate a cardioloid from different pattern sizes. You can see we can generate a cardioloid from 200 microns patterns, 600 micron patterns, and 1,000 micron patterns. We're using a GCAN reporter iPSC line, so we can track the calcium transit of the cardioloid as an indication of the electrical physiology of the cardioloid. That will help us to understand the contractile function of the cardioloid. So with this technology, so we can actually generate macro patterns with so many different shapes. And then what we want to understand is how this geometrical control or geometrical confinement can regulate cardioloid functions. So what we do is we generate 3 major functions, circular functions with different area size, right? 200 microns, 600 microns, and 1,000 microns. And then we generate this like more complicated shapes, 2 star shapes and 2 quantilateral shape, square and the rectangles. The geometrical area for the star shapes and the rectangle and the square shape is the same size as the 600 micron circles. And what we do is just macro pattern the iPSCs, induce the differentiations, get the cardioloid, quantify their functions. So in terms of the quantification of the functions, we go with 2 major technologies. The first one is we image the cardioloid in the bright fields, and we record beating videos, quantify the contractile motions. And the second, we switch to the GFP and record the fluorescence to quantify the calcium transit. And from each video recording, we can quantify the motion, contractile motions, and the calcium transit that give us 10 key sort of like a physiological parameters. And then what we get here is we have there's 7 different geometries. Circles, rectangles, squares, and 2 star shapes. Each geometries we generate a decent number of the cardioloid. In total, we have about like 230 cardioloid. It's not huge numbers that you can input in large language models, but we try to find a way using a more simple machine learning models to help us to understand the physiological function of the cardioloid. From every single cardioloid, we can have 10 physiological parameters, 4 from the contractile motion analysis, and 6 from the calcium transit analysis. And so this is our data set, and then we input this into 2 different machine learning pipeline. One is unsupervised learning approach using dimension reduction techniques and clustering techniques to understand the relationship of the cardioloid from different geometries. And the second, we're using machine learning classifier to help us to see whether we can have better prediction the cardioloid from different geometries. So you have known about the UMAP, right? So basically, it's tell you that the spatial relationship between the cardioloid with different geometries. Each dot is the one cardioloid integrated with 10 different physiological parameters. And you probably can see there's some cardioloids was separated far away from the major cardioloid populations. That I think this is the laser pointer. That is a 200 micron cardioloid, which is small little size, have very unique functions. If you see the functions of the 200 microns, you can see the cardioloids was clustered into cluster 4. And if you go back to the cluster 4, you can see what's the unique function of the 200 microns cardioloid. This helps us to really identify whether we can have all the cardioloids was populating in the whole populations, and then understand what's the functionality of specific clusters that was identified by the machine learning techniques. But in terms of the dimension reduction method, they're not just UMAP. There's so many different dimension reduction methods. So like Disney, they really focus on the separate local similarity between the cardiloid, all the way to the PAC-map. It's promote a more global view of the relationship between the cardiloid from different geometries. And particularly from the PAC-map, you can see there's a progression from a tiny, small 200 microns cardiloid progress to the more like a star shape, and then larger shape with rectangles and the 600 microns circles. And you can see from this dimension reduction techniques, the functionality of the cardiloid was a progress change from one end of the map to the other end of the map, showing the progression from small size of the cardiloid to the larger size of the cardiloid. The technique's cool, right? But what we eventually get is 4 different dimension reduction techniques, and each one will produce 7 different clusters of the cardiloid. Total, we have 28 clusters. And then it's very difficult to understand the relationship between so many different dimension reduction techniques and clusters. So what we do is we perform this network analysis between the clusters that came from different dimension reduction techniques. The network analysis has been used a lot in the social networks. So if you have a friends, and there's another people have a common friends, and you and the other people was linked by the common friends. Similar here, if one cluster from one dimension reduction techniques have a coexisting organoids from the other dimension reduction techniques, they will link each other's. But that doesn't help us too much unless we can refine our organoids populations. So what we do is we regroup all the organoid clusters from different dimension reductions into one communities. It's like we have our own communities, right? You have a communities at work with your colleague, and you also have a community in your neighborhood with your neighbors. So, but your is the link between all these communities. So what we eventually see is we create these 6 different communities. The communities for 200 microns cardiloid, or the communities for like rectangle 1 to 4 cardiloid. And then what's in between the communities is the cardiloid that was shared by different communities. That's where the cardiloids have higher heterogeneities. So what we do is actually we want to refine our organoid communities by proving all the connections between the communities to refine our cardiloid populations. So you probably can see from 200 microns, right, we didn't get anything proved because it created its own unique communities. But for the star 1 to 1, we actually have almost 60% organoids approved to further refine the organoids that can better represent the star 1 to 1 shapes. At the same time, I want to highlight this rectangle 1 to 4. You only got like a 10% reduction, which indicate there's a better homogeneities or reproducibilities from the cardiloid from this rectangle 1 to 4 geometries. And the reason I want to highlight this rectangle 1 to 4 geometry is because as a cardiac physiologist training background, we can see this rectangle 1 to 4 have a better contractile functions compared to all the other cardiloid, which indicate this rectangle 1 to 4 geometry can enhance the physiological function of the cardiloid. So, and then, as I say, we have another pipeline to using machine learning classifier to try to predict which organoids came from which geometries. We trained 2 different models, this random forest and a gradient boost, to see, okay, if I give you an organoid, can the machine tell me which geometry this organoid came from? And you can see from the testing data set, so we have about 60% prediction about each the organoids came from specific geometries. The similar way also can do the prediction from the organoids that was belong to each cluster of the different dimension reductions, and you can the prediction for the organoid cluster from different dimension reductions is much higher, just because what is learned is based on the clusters. And the uniqueness of using this random forest or the gradient boost, this assemble approach for machine learning classifiers, it can help to understand which feature is most important to help you to help the machine to do the to do the classifications. So you can see this causing rising time, which is how much time take the cardioloids to uptake the causing and start the contractions. That's the feature the machine use a lot to make the predictions, which indicate that's the most important feature to classify the cardioloid from different geometries. And what we see from previous analysis, the rectangle 1 to 4 has the very low causing rising time compared to all the other organoids, which indicate that rectangle 1 to 4 have these improved functions compared to all the other organoids. So what we do is take this rectangle 1 to 4 and do the immunostainings and to verify like what we can see from experimentally can validate the machine learning models. And what we do is just do the three-dimensional mapping of the cardioloid from 600 microns and the rectangle 1 to 4 micron, 1 to 4 shape of the cardioloid. What we see that we do the staining for the sarcomeres and then image use the confocal microscopies. We didn't see a significant difference in terms of sarcomere length, but what we see the sarcomere alignment for the rectangle 1 to 4 cardioloid is significantly improved. However, the overall three-dimensional morphology is relatively smaller for the rectangle 1 to 4 cardioloid compared to the 600 micron cardioloid. So next, the similar, we take this two different cardioloid and run the bulk RNA sequencings. And we see that the transcriptome-wise, these two, the cardioloid from different geometries have quite different transcriptome profiles. And what we really see is the rectangle 1 to 4 organoids have a better cardiac muscle development, higher causing signaling pathways, and also the muscle contraction signalings. So basically, our bulk RNA sequencing data and also staining data verify that the rectangle 1 to 4 cardioloid had better functions and the cardiac development compared to the 600 micron circular cardioloid. So we're quite happy our result. We send the manuscript for review, and then the reviewer asks, is your model generalizable, or whether you can reproduce your data? So what we do is after almost 2 years to generate the first batch of the cardioloid data, we generate our second batch of the cardioloid data just for the rectangle 1 to 4 and the 600 microns. And what we do is we collect all the 10 physiological parameters and we project them back to the UMAP we generate based on the original data. And that's showing as the green colors here. And so you can see we still, the new cardioloid still be able to project back to the same UMAP with the similarities. And then we also ran the classifier to see whether we can predict the new rectangle 1 to 4 is still the rectangle 1 to 4 based on the machine learning model was trained by before. And we can see that the accuracies of course, dropped to about 60% compared to 70%. But what we really see that the rectangles precisions is significantly higher than the circular shapes. And so that will tell us is the reproducibility to produce the rectangle 1 to 4 cardiloid, which is about 70% or 90% based on different models, is much better than the reproducibility of the circular cardiloid patterns that we can generate our cardiloid. So with that, we sort of are building our machine learning pipelines that can help us to optimize our cardiloid productions. So we already have this 7 different geometries. It's the first generation of the cardiloid libraries. We go through these machine learning pipelines, and we can further refine our cardiloid to highlight the unique functions from different cardiloid with different geometrical patterns. And then we identify that this rectangle 1 to 4 have enhanced the functionality, but as we see in the confocal microscopy, the overall morphology and the 3D volume is decreased. So what we plan to do is to integrate the circular patterns and the rectangle patterns to create more complex geometries that can so we can have enhanced physiological functions, but at the same time we can have a larger volume of the cardiloid. And we can feed this new cardiloid into our cardiloid libraries to create this more like optimization loop to better design the cardiloid for future use. And to summarize, we established these cardiloid models. We're using geometry confinement to control the functions, and then we're also using machine learning to understand their functionalities. And thank you so much, and happy to take questions.
We have time for questions. Maybe one question for Dr. Ma first, then you can ask questions for all the speakers. Okay.
This is kind of a very basic question. But when you look at the various geometrical shapes you tested and you look at the functionality, from an intuitive approach, do they, in a sense, make sense that a certain geometrical shape would lead to such and such a functionality just based on what you know from in vivo systems? Any thoughts on that?
Think about like a mouse heart and a human heart, right? The total volume is quite different, right? How much force was generated to pumping the blood is different. And think about the simple beat rate, right? Mouse heart is very small, it beat 100 beats per minute, and human heart about 60 beats per minute, right? So somehow this overall geometry and especially the size or the volume make a difference in terms of the physiology and the function output. And from in vitro analysis, right, if we were only looking at like 3 simple size, 200, 600, and 1000, it's actually the middle size, the 600 have a larger volume. That's the reason we use for a lot of like Barker-seq and the immunostaining analysis. But when you further increase the size, 1,000 microns, the creation of the cardiloid was now as robust as the 600 microns, and especially they don't pile up to form the 3-dimensional morphologies as the 600 microns. When you have a larger size, the differentiation and the morphological change in vitro is more like a 2D differentiation that normally we can do in the 6-well plate. So how you can control the geometry definitely can change not just the functions, but also the morphology and the volume that's important for the heart.
Great, thanks so much.
Good morning, everyone. My question is more so geared towards Christy Taylor. I am Sade Day, a student at Tuskegee University, and my question is, in the context of accelerating product development in vitro biology, how do you see AI helping to bridge the gap between highly controlled lab-based optimization and the complex variability of real-world agricultural environments? And then also, how can we better communicate these AI-driven advancements to farmers and stakeholders to build more of a trust and understanding?
Okay, I'll try to answer part of that. So as far as in vitro biology goes, I think AI is driven by experimental data, right? And the only way we're going to get enough experimental data to drive these models is through in vitro biology, to be honest. So I see this as an incredibly important venue to drive new experiments, new ways of testing things that we can feed into these models. I foresee us being able to put in many different types of in vitro data in the future to drive these models forward as well. If you want to, it was kind of a long question too, so if you want to give me the last part of that, or is there I think you said something about acceptance as well.
Yeah, like communicate, like how can we better communicate these advancements to basically like the general population to get them like more on board?
Yeah, I think that AI can seem kind of scary to people right now. We are also, at least the OpenFold Consortium, I'm going to speak based on that. We're involved in a lot of biosafety discussions as well. There's folks like Mohammed Al-Qureshi with whom we collaborate. He's been called to Washington, D.C. to talk about those panels. So it's very much on the minds of people to make sure we're still advancing products forward, but doing it in a responsible and safe way. Right now, the way that people are looking at that is through, for instance, the DNA synthesis companies, for instance, to make sure that people aren't doing things inappropriately. But also thinking about that one of the advantages, too, of having open source is we're able to see what is actually in those training sets versus these closed models where we have no idea what they're trained on. So I think the open source aspect is also very important. So hopefully that answers your question.
Yeah, thank you.
All right, thanks.
We can have one more question if there's one. So if not, I think we are on the time. So please join us to give a round of applause for all our speakers. And thank you very much for your attending.